of this chapter is about DNA sequencing. So DNA sequencing is used to determine the nucleotide sequence of a specific DNA fragment or the nucleotide sequence of a whole genome. Okay, and in this chapter we're going to learn about the newest sequencing technique, because there were there have been significant improvements and developments in the DNA sequencing techniques or methods in the last 15 years. The first automated procedure that was used for sequencing was called the dideoxy chain termination sequencing, or sometimes it's called Sanger sequencing. Sanger is the name of the scientist who discovered or who developed this sequencing technique. Now, sequencing could be done inside machines, and these machines are automated machines. Okay. Now, currently we have what we call, let's say we have old sequencing techniques. We have also new ones, or what we call next generation or next gen sequencing technique, الجيل الجديد of the sequencing techniques. So new techniques. Okay. So in this course, I'm not going to spend a lot of time explaining how the old techniques were done, because you will learn about these sequencing techniques in details in the biotechnology course. So in the biotechnology course, you will learn, so BT 230, basic biotechnology. So in this course, we typically teach students about the details of several types of old and new techniques. So next generation sequencing techniques consist of many sequencing techniques developed in the last, let's say, 20 years. Your book lists one of these techniques. And in this technique begins, or sequencing begins, by having a genomic DNA. So these next generation sequencing techniques, they could be used to sequence all genomes. Okay. So I'm going to just explain sequencing technique that is mentioned in your book, your textbook, and you will learn about others, inshallah, in the biotechnology course, and about other techniques in the biotechnology course. So let's suppose that we want to sequence a genome inside cells. Of course, here we're just showing you one cell and one genome, but actually we will begin with millions of cells and millions of identical genomes. These genomes first are fragmented into smaller fragments. Now, each fragment then will be isolated with a microscopic bead. Bead means kharaze. Okay. Then, on the surface of the bead, PCR will be used to make more identical copies of each fragment on a bead. So here, what you see, here we have a bead. On its surface, there will be 10 to the power 6. So this is 1 million copies. All these copies, 1 million identical, they will have so each bead will have on its surface millions of copies of identical DNA fragments. So all these DNA fragments you see here, they all have the same sequence. Of course, here just showing you one fragment on one bead, but actually there will be millions of beads carrying millions or billions of DNA fragments. Okay. Then what will happen is that each bead carrying these millions of identical DNA fragments, these are short DNA fragments, each bead will be placed inside a well. So there will be millions of wells. Here we're just showing you a couple of them. Each well will have one bead because we start with millions of beads. We will have millions of wells. Each well will have only one bead. What will happen if we zoom in, as you can see here, this is a DNA that is found on the surface of the bead. Of course, we have millions of copies of this DNA on the surface of each bead. Now, inside the well, we have a primer that will bind to these DNA fragments on the bead. And we have also DNA polymerase. Okay. So the DNA polymerase will come here. So this is the DNA polymerase. And it will start synthesizing a new strand from this, using this template DNA strand. But the synthesis will not happen continuously. It will happen as pulses of synthesis, which means there will be no, the 4 nucleotides that are used to synthesize this strand will not be available all the time for the DNA polymerase. These nucleotides will be added one at a time. How? So the DNA polymerase will bind to the primer. And now here we have something similar to the PCR. No, here we have DNA primer. This is the DNA primer. So all these DNA fragments on the surface of the bead will be bound by the DNA primer. And as you can see here, we have this is the DNA polymerase. Now what will happen, we have the 3 here, we have the DNTPs, the deoxynucleotides, the A, the T, and the C, and the G and the C. And these nucleotides are separated from each other. Okay? And these nucleotides will be added one at a time, will be added sequentially. So what will happen is that A will be added to all wells first. Then after adding A will be removed from all wells, and then T. After adding T, all T's will be removed from all wells, and then G will be added, and so on. And this process is repeated over and over. So basically, inside the wells, you will not find the 4 nucleotides at the same time. Inside the well, A is first added, then A will be removed, then T will be added, then T will be removed, then G will be added, then G will be removed or washed off, and finally C, and then go back to E. So this is the direction of addition. Then we go back like this. So what will happen is that, imagine if this is the template DNA and this is the primer. Okay, so the primer is from here to here. On the template DNA, after the primer we have T, the DNA polymerase will bind. The first nucleotide that should be added to the newly synthesized strand should be A, because on the template here we have T. So if we add A first, okay, there is A, and A is needed here. The DNA polymerase will add the A. You have to understand that we are using DNTPs. I told you N could be the nitrogenous bases A, C, T, or G. T, P stands for, so the T, so D means deoxy, because we're talking about, so deoxynucleotides, because we're talking about DNA. The N is any of the nitrogenous bases. TP, triphosphate. Tri means 3 phosphates. So we have the nucleotide, let's say we have nitrogenous bases. Then we have deoxyribose sugar. So here we have H, OH. Then we have 3 phosphates. Okay, so this is the end. Now, these are the nucleotides that are found here. In here we have, instead of an A, in here we have T. When these are available for the polymerase, the polymerase will add a nucleotide and removes 2 phosphates, okay, from the nucleotide. So these are called pyrophosphate. If we add to the well nucleotide A, and if A is added, A will be added, 2 phosphates will be released, and the 2 phosphates will be used to produce a color or a light, and this light will be detected. Okay, so after the addition of A, because A was incorporated or added to the newly synthesized strand, pyrophosphates are produced, and these pyrophosphates will be used to cause a flash of light or to produce a light. Light is produced if, only if, the nucleotide is added by the DNA polymerase into the newly synthesized strand. Remember, we add A, okay? And when we add A, not all wells will incorporate A into their newly synthesized strands by the DNA polymerase. Only some wells. So maybe we will have light produced here. Okay, let me use the color green here. Maybe it will be produced here, 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 and here. So basically, after adding A, so we added A, color will be produced only in these wells, which means these wells, okay, in these wells, the DNA polymerase added the nucleotide A as the first nucleotide into the newly synthesized strain and produced pyrophosphate, and pyrophosphate was utilized to produce light. Okay. So inside the sequencing machine, there is a scanner or a camera that will record which wells produced light after the addition of A. So A will be added to all wells, but only, let's say, only few. Well, let me use. So the green color is misleading. Let's say that the color is just let me use this color. Let's suppose that this is the color that is produced after the addition of A. Okay. Now, after A is added, some wells will produce flash of light. There is inside the machine a camera that will record which wells produce the light. After that, A will be removed and T is added. Okay, of course, the light will be, it's a flash of light, so the light will go away. And after adding T, other wells will produce light. The camera will record which wells added, produced light after the addition of T. Then we remove T and the light is now gone, and we add, let's say, G. So we have, let's say, G added. And again, we have some wells that will be able to incorporate or add G nucleotides in the newly synthesized strand by the DNA polymerase and produce pyrophosphate, which will produce light. The camera will record the light. And then G is removed. Then we have C added. And then again, we have some welds will produce light, and these welds will be recorded by the camera. Okay. And then again, C will be removed, and we go back into adding A again. And this time also, the welds that will produce light are the welds that incorporated A. And this process is repeated many, many times. So in this example, after addition of A, this is just, we are looking at one well. Okay. This is the template DNA on this well. Because the first nucleotide that should be added by the DNA polymerase happened to be A, and we provided this well with A was added, pyrophosphate was released, flash of light. The next nucleotide to be added is T. T will be added after removing A, so A will be removed, and T is added. Notice that the DNA polymerase does not need a T here. It needs a C, not a T. But we give the polymerase a T, so the T will not be added. No color will be produced. But the next nucleotide that will be added is G. And again, we need here, or the DNA polymerase needs a C. But we added for the DNA polymerase inside the G. The G will not be added. No color will be produced. So after adding the C is needed. It will be incorporated, pyrophosphate will be released, a flash of light will be produced. So basically, inside the machine, we have a sequential addition of nucleotides that will cause some welds to produce light. There is a camera that will record which welds will produce light after the addition of which nucleotides. And while this is happening, a newly synthesized strand is being made, and at the same time the machine will know which amino acid, which nucleotides are being added to the newly synthesized strand. So here we have synthesis of newly synthesized strand and sequencing of the newly synthesized strand at the same time. Okay. Yes, all these DNA sequences that you see here, all these DNA fragments, they will have the same sequence. All of them will add A first. All of them will not add T after adding T. All of them will not add G. And I'm talking about the DNA fragments in the same well and on the same beat. All of them will add the second nucleotide as C. Then after C, we will go back to A. So if we continue here, let's say if we want to continue. So remember, in the first cycle of addition, we add A, T, C, G. So we added A, then T, then G, then C. Next we'll be adding what? A. Do you think after adding A, there will be a light produced or not? Look at the template sequence. So after adding C, in the template DNA, what? We have T. So if we add A is needed here, so there will be light. Yes. Okay. And then after that, we have to add what? After A, we have to add T. Do you think after adding T, there will be light? Yes, because here we need two T's actually. So the light that is produced will be even what? Stronger light, because two T's are added here and two pyrophosphates will be produced here. So the light will be stronger. And then if we continue, and the next step is to add a G. Do you think adding G will produce light? Yes, twice the amount of light actually. And this process continues and continues like this. So adding one nucleotide at a time might produce light and it might not produce light. Okay. So this is how the result look like. So here we have a cycle of adding nucleotides in the following order. So in this example, the first nucleotide added is the T, followed by the A, then the C, then the G. Okay. Here the sequence is different than the one shown in this figure. So as you can see here, after adding T, okay, this line represents corresponds to the number of nucleotides added in the newly synthesized strand. So here we have 1. So after adding T, there was a light, and this light corresponded to the addition of 1 nucleotide. Then after adding A, there was no light. After adding C, there was a light. After adding G, there was no light. After adding again A, no light. After adding T, no light. After adding T, there was a light that corresponded to the addition of a single nucleotide. So if I look at this figure, I can determine the sequence. So after adding T, so here we have another T, then followed by A, then here we have G, then here we have C, and this is continue. T A C G, T A C. T A. Well, I made a mistake. This is G. This is a C. T A C G, T A C G, T A C G. Okay, and each nucleotide might not produce a light, or it might produce a light, or it might produce a stronger light that corresponds to the addition of 2 nucleotides. So by looking at this, we can determine the sequence like this. Let me show you how we can read this figure and determine the sequence. So after adding a T, there was a light. The amount of light produced corresponded. By the way, this is the result of 1 bead on 1 well. Okay, 1 well, 1 bead. Remember, we have millions of beads carrying. Each bead will be carrying different DNA fragments. So after the addition of T, there was a signal. So the first nucleotide added was T. After the addition of A, no signal. After the addition of C, we have a signal. So the second nucleotide added was C. After adding G, no light. After adding T, again, no light. After adding A, there was a light. After adding C, no light. After adding G, there was a light. After adding T, no light. After adding A, no light. After adding C, we have a light. After adding G, we have a light. After adding T, we have a light. After adding A, we have strong light that corresponds to the addition of 2 nucleotides. So here we're going to write AA. After adding C, no light. After adding G, we have a light. So double the amount of light. We have to write here GG. Then we have T, no light, A, no light, C. Then we have G. We have no light T, light A. And this will continue. Let me write it here. T, this T. Here we have this G. Here we have this T again. Here we have this A. Here we have this T. Here we have this A. And then notice here, we have very strong light produced after the addition of G. And this strong light corresponds to the addition of 4 Gs. G, 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 G. Okay. And we could continue to the rest of, of this figure. Okay. Does this process take a long time? No. Actually, it takes, doesn't take that much. Okay. طيب دكتور ممكن نستخدمه في الوسط كلاسي. Yes. Okay. Assalamu alaikum, everyone. So far in this chapter, we discussed gene cloning, or in general DNA cloning. Okay. And we learned how DNA cloning could be used for many purposes or many applications, like just making more copies of a gene, or maybe sequencing a gene, or maybe study a gene sequence, or sometimes to express the gene. And also we learned about the PCR as a way of making also more copies of a DNA fragment. Also, PCR could be used for gene cloning. We learned about expression system. Expression systems involves cloning a gene and inserting that gene into a vector and inserting into an expression vector and inserting the expression vector carrying your gene into a host expression system that will transcribe and translate your protein. Also, we learned in the previous lecture about DNA sequencing, about one of the next generation technique of DNA sequencing. All of these techniques or methods to analyze or to study DNA will be discussed in details, inshallah, in the biotechnology course BT 230. Today, inshallah, we are going to learn about what we call gene expression techniques, the techniques that will allow us to study gene expression, to determine which genes are expressed, which genes are not expressed, and the level of expression, is it high or low? Okay. All right. So one way of, knowing more about a gene or its function is to understand when and where, okay, a gene or a group of genes are expressed. So when means, let's say, what stage of the organism's life, or maybe at what, let's say, conditions, or in what conditions. So when a gene expressed, sometimes some genes, as we saw in the previous chapter, they are expressed during development, and later on they are not expressed. And some genes, they are not expressed during development or during embryo development, and then they are only expressed at the adult stage. In what conditions means that, just like we learned in the previous chapter about how gene expression could be coordinated in response to, for example, the binding of a hormone or a growth factor on the surface of the cell. Okay. So when means maybe a gene will be expressed at a specific developmental stage or at a specific situation during the life of the organism. Where here means we are interested in knowing which cells or tissues express the protein. Okay. And there are different techniques that could be used to answer these questions, when and where a gene is expressed. And sometimes we might be studying a single gene, or maybe studying the expression of a group of genes or a larger number of genes at the same time. So typically, most of these techniques will focus on detecting and identifying the mRNA. Remember, gene expression for most genes starts with the DNA that is being transcribed into mRNA, and then we have translation of the mRNA into a protein. Okay, so here we have transcription, and here we have translation. Now, if we could identify the presence of mRNA for a specific gene, this means that probably the gene is being expressed. If we find an mRNA for a specific gene inside the cytoplasm of cells, this means that the gene is being expressed. Sometimes also we could be looking at the protein. Okay, but most of the techniques that we will be discussing in this course will be looking at the mRNA, and not only the presence or the absence of the mRNA. So not only presence or absence. Presence or absence means موجود or not, but also we could look at the mRNA level. So we have let's say we have a qualitative measurement of the mRNA or assessment of the mRNA for a specific gene, and we have also quantitative. Okay, qualitative means وصفي. Is it present or not? If it's present, the gene encoding the mRNA is expressed. If it's absent, this means the gene is not being expressed. This is a qualitative assessment of gene expression. We have a quantitative assessment, كمي. Okay. This is to determine whether the gene is expressed. So the gene is expressed, but the mRNA, the amount of mRNA we find inside the cell, will determine whether the gene is expressed at a high rate or at a low rate, or there is a high level of expression or low level of expression. Okay, same thing applies to protein. But again, we are going to focus in this chapter about the techniques that will detect the presence, absence of mRNA and the level of mRNA, the mRNA levels that are found inside the cell or detected inside the cell. Now, most of the techniques so far we have been discussing, they depend on the principle of nucleic acid hybridization. Okay, nucleic acid, this is DNA or RNA. Hybridization here means we have one DNA molecule, let's say a DNA molecule, will be converted into two single-stranded DNA molecules. And a third DNA molecule or a single-stranded DNA molecule could hybridize to any of these. So here we have hybridization between, let's say, this is a double-stranded DNA molecule. Okay, so DS means double-stranded. Okay, and here we have a single-stranded, let's say, DNA molecule, and this single-stranded DNA molecule could be complementary to one of the strands. So this is a different DNA molecule, single-stranded DNA molecule that is different, that has a complementary sequence to one of the strands of the double-stranded DNA molecule. Or it could be an RNA, for example, single-stranded RNA molecule that will bind to this DNA molecule. So this is what is meant by hybridization. Hybridization, we have the binding of two nucleic acids to each other. This is what is meant by hybridization. And we will learn how this hybridization is the basis of many of the techniques that are used to detect gene expression. So this, for example, is a DNA hybrid. Hybrid gene means. So this is a DNA hybrid. Hybrid means a gene because we have the hybridization of two different DNA molecules from two different sources. Okay, or we have here RNA DNA hybrid. Okay, so the first technique we're going to discuss depends on the use of nucleic acid probes. Okay, this could be a probe. Okay, and this could be a probe. The probe could be a single-stranded DNA or RNA molecule that is complementary to our target sequence of our choice, and it is used for the detection of a specific DNA molecule. Okay, many of the gene expression techniques, they rely on the use of nucleic acid probes. These probes typically they have extra things they are carrying with that will allow us to see or to detect the presence of a specific target sequence. So for example, if we have, let's say, a long DNA molecule, And we are interested in detecting the DNA that here. This is the DNA target. We are interested in detecting. Okay. We could use a DNA probe. Okay. So this is a DNA probe. And the sequence of this DNA probe should be complementary to the DNA sequence we are looking for in the genome. So this could be, let's say, a whole genome, all the DNA inside, let's say, a cell. And the probe is carrying with it, let's say, a fluorescent dye, a dye that will give a fluorescent color, upon the binding of the probe to the DNA. So the probe will come and bind here, okay, if the two strands are separated from each other. So the probe will tell us whether our target DNA is found in this whole genome or not. Okay, this is the basic principle behind using probes to detect specific DNA sequences or specific target sequences. Now let's see how probes could be used for different techniques. The first technique that we're going to discuss is called in situ hybridization. Okay, in situ means in site or in its native site. Site means mocha, native means asli. So here, this technique will be used to detect the presence of, remember, we're talking about gene expression. So we are trying to detect the presence of mRNA. Here, this technique is called in situ hybridization because we are trying to identify the specific mRNAs in their native site, in their native location, in the location in which type of cells or tissues the mRNA will be present. And hybridization, because this technique uses a probe, and this probe is carrying a fluorescent dye that will tell us where this mRNA, and sometimes when, will be found inside specific types of cells or tissues. So imagine if this is our target sequence. This is an mRNA that we are looking for. This mRNA corresponds to a gene. So if we find this sequence of mRNA inside a cell, this means that the gene encoding this cell is expressed. So we're talking about a gene that is being converted into an mRNA, and we want to see if we can detect this mRNA inside a cell or not. Now, in order to do so, we use a probe. So this is a DNA probe because it is made of DNA. But notice we are looking for an mRNA, but that's okay. So this DNA, okay, will be complementary to this RNA. Okay, so remember, the probe that we are going to use has this sequence, let's say, which is complementary to this RNA. So I'm going to write it here: G, okay, A, G, T, A, G, T, G, G, C, C, G. So this is the probe. This is a DNA probe that will bind to a complementary mRNA that we are looking for. And notice that I used the yellow color to indicate, I used to write yellow color for this probe to indicate that this probe is a fluorescent probe. Okay, so this is a fluorescent. Do you know what fluorescent means? Phosphory. So it will give a fluorescent yellow color if this probe binds to its target DNA. Okay. Yes, ya Aziza, you have to know the sequence of the mRNA you are looking for. That's why you have to design a probe that has a complementary sequence to the sequence of the mRNA you are looking for. Yes. You have to have knowledge about the sequence of your gene, maybe, or the mRNA encoded by your gene. Okay. So, in this technique, in this figure, for example, this is Drosophila larvae. And here, as you can see, we have two mRNA molecules, or two target mRNA molecules. And each one of these mRNA molecules has a specific, let's say, a specific DNA specific sequence. And we are trying to look for both mRNAs using two different probes. So this blue probe has a sequence that is complementary to this mRNA. This yellow probe, okay, has a sequence that is complementary to this mRNA. So we have two mRNAs, EN mRNA and WG mRNA. You don't need to know what these two letters means. All you have to know is that we have two genes in Drosophila called WG gene, and EN gene, and these are the two mRNAs produced by these two genes, or encoded by these two genes. Now we want to see which cells inside the Drosophila larvae will be expressing the EN gene and which is expressing the WG gene. To answer this question, we need to use probes that are complementary to both mRNAs encoded by both genes. And notice here, we have, this is what is called in situ hybridization. So the larvae, okay, the larvae cells. So we will add these probes to the larvae cells, and these probes, once they find their target sequence, once they bind to their target sequence, they will produce these colors. So wherever you have a yellow here, yellow means these are the cells in which we found the WG mRNA, which means these yellow cells, they are expressing the WG gene. And then we have cells that are colored blue, indicating that our blue probe, found its target complementary mRNA, which means that these cells are expressing the AN gene. No, these probes, they are synthesized in the lab, and we introduce them into the inside of each cell within the larvae. And notice here, we have also maybe red cells to indicate that these cells may be expressing or indicates the presence of, let's say, XY mRNA. Okay, using red probe, maybe a third probe. Okay, we have purple cells, which means that these cells are expressing maybe, let's say, RT mRNA, or they found an mRNA called RT using a purple probe. Okay, so these are just examples. So each one of these probes, whether it's a blue, let's say, yellow, purple, red, each one will be looking for a different gene. And we have also here, we have green probe, or probe with a fluorescent green dye, looking for, let's say, ZY mRNA. So each one of these 5 probes will be targeting or will be complementary to a different mRNA molecule. Okay. But notice here, if we zoom in, we have also, so let me draw a line to delineate the body of the larvae. So this is the body of the larvae. Okay. And notice here, we have these black regions. These black regions does not mean that we don't have cells here. No, we have cells. But the cells here, which we cannot see, do not express any of the mRNAs for which we use the probes. So the cells that are found here, okay, let's say here we have cells, these black regions. These cells, they do not express any of the mRNAs, any of these mRNAs that we are trying to detect. The XY, the RT, the ZY, the EN, the WG. This is why there will be no probe binding into any mRNA inside these cells. Okay, so here we could say I could draw a line like this. No expression of, let's say, WG, EN, ZY, RT, or XY mRNAs, or genes, let's say, genes. And if we zoom in more, notice here we have purple and blue, indicating that these cells might be expressing the EN gene and the RT gene at the same time. So here we have cells that are producing both purple and blue colors, which means both probes, the purple probe and the blue probe, are binding to mRNAs inside these cells. Okay. And notice here also, if you look at this red line, we have cells that are producing intense red color, and some cells here, they are producing some red color. This means that the cells in the middle here, you see, they are expressing the XY gene at a higher rate than the cells that are expressing also the XY. So here we have high level of expression, which is indicated by the intense red color. Here and here, for example, and here and here and here, around this line, red line, we have still some red dots, indicating that the cells here, here, they are expressing XY gene, but not at a level that is comparable to these cells. So here we have high level of expression of XY. Here we have low level of expression of the XY gene. Okay. So as you can see from this technique, we can detect where a specific gene or a group of genes within an organism, within tissues, are being expressed. So in the head, as you can see, we have yellow, WG, mRNA. And again, this technique is called in situ because we are detecting the mRNA inside their location inside cells. Okay. Why do we specifically insert DNA, single-strand DNA, not single-strand RNA? You could use single-strand RNA, but DNA, let's say it's more stable. Okay. It's easier to use it as a probe compared to RNA. But in theory, yes, you could use RNA. That's fine. Another technique that is used to study gene expression is based on the polymerase chain reaction. It is similar to the polymerase chain reaction you learned about in the previous section, but here it's called reverse transcriptase polymerase chain reaction, or RT-PCR. This technique, in this technique, additional enzyme called reverse transcriptase is used. So let me remind you about polymerase chain reaction. In polymerase chain reaction, in PCR, we use a DNA template that contains the DNA sequence that we want to amplify. We use DNTPs, nucleotides, the 4 kinds, A, C, T, and G. These will be the building blocks for the newly synthesized DNA molecule. We use also DNA polymerase. We use also primers to determine which DNA sequence will be amplified. And then finally, of course, a buffering system that will provide the polymerase with the proper chemical conditions to carry out its function. Now, in RT-PCR, the template is not a DNA template, it's an mRNA template. We also use DNTPs, just like PCR. We use also DNA polymerase. We use also primers, a buffering system. And then we have to use an extra enzyme called reverse transcriptase. So the DNA polymerases, they use DNA template to make more DNA molecule. Reverse transcriptase uses RNA as a template to make DNA. Okay, so it's like PCR instead of using DNA as a template, here we're looking at the use of mRNA as a template. And also we have to use a reverse transcriptase enzyme that will convert the RNA. It's called reverse transcriptase because it will, typically in transcription, we convert DNA codons into RNA codons. Here, reverse transcriptase, this enzyme carries out reverse transcription. So it will carry transcription بالعكس. It will convert RNA into DNA. Okay, so this is the opposite of transcription. Transcription usually goes from DNA to RNA. Here we have reverse transcription from RNA to DNA. That's why it's called reverse transcriptase. Now, the idea behind using RT-PCR to study gene expression is if the mRNA corresponding to a specific gene is found in the cytoplasm, and we use that mRNA to do PCR. If the PCR successfully make DNA copies of that mRNA, this means the gene encoding that mRNA is expressed. So we use PCR amplification to detect the presence of specific mRNAs inside the cell. Okay, so this is how the RT-PCR works. We have mRNA inside, let's say, inside the cell. And we isolate these mRNAs. We don't start with DNA, just like in regular PCR. We isolate mRNA in a tube. Then into the tube, okay, we add everything from 1 to 6. So we add, from 2 to 6. We add DNA polymerase. We add primers. We add DNTPs. We add reverse transcriptase. And we add a buffering system. And then what will happen? First, the reverse transcriptase will convert all mRNAs into, it will use the mRNA molecules as a template to synthesize a DNA strand. Okay. The reverse transcriptase also, just like the DNA polymerase, synthesizes in the direction from 5' to 3. Here, because all mRNAs inside eukaryotic cytoplasm, they have all poly A tail, we could use poly T primer. So this will ensure the use of poly T primer will ensure the amplification of will ensure that the newly synthesized DNA strands from these mRNAs will be produced. Then the mRNA, after being used as a template, will be degraded, will be removed. Okay. And then the DNA polymerase will use the DNA template to synthesize a double-stranded DNA molecule. And this double-stranded DNA molecule is now called cDNA. C stands for complementary. So we converted an mRNA, a single-stranded RNA molecule, into a cDNA, which is a double-stranded DNA molecule. Okay. And now this cDNA, yes, ya Aziza, does not contain any introns. It's a DNA of the gene that without the introns. Okay. Because remember, the mRNA we have is basically produced from a gene that consists of exons, if we're talking about prokaryotes, and introns. And through transcription and RNA processing, the gene codons will be converted into mRNA codons. Remember, all the mRNAs in the cytoplasm of eukaryotic cells, they don't have introns, they have exon codons. Okay, so now this is like it's like converting a gene. Let's say that we have this gene, okay, consisting of both introns and exons. So this is intron, this is intron, this is exon 1, exon 2, exon 3, then again transcription, RNA processing. We have what? We have mRNA. Okay. And then using reverse RT-PCR, we will end up with a double-stranded DNA consisting of exon 1, exon 2, and exon 3. Okay. This is the cDNA of this gene. Now, let me stop here, and inshallah we'll continue after 5 minutes. Let's take a 5 minutes break. Okay, so now, since we learned about the principle of RT-PCR, and we learned about PCR and generic freezes in previous section, let's look at an example of a way of detecting expression. Let's say gene expression of a number of genes. Let's say that we have a cell, and this cell contains, let's say, this cell cytoplasm contains the mRNA of different genes, and we are interested in detecting the mRNA of gene T, purple, gene Y, yellow, and gene B, blue. Okay. So what we will do first is, so this is a nucleus. We will first extract mRNA, all mRNAs, and we add all mRNAs into 3 different tubes. Okay. So this is tube 1. This is tube 2, and this is tube 3. So in each tube, we will add the mRNA for the total mRNA extract, let's say here, total mRNA, all the mRNA inside the cell. And inside each tube, we add fraction of this total mRNA. So we'll have mRNA for the blue, some mRNA from the purple, and the yellow. And to each tube, so here I'm going to write to each tube, we add the RNA, the RT-PCR, the reverse transcriptase, we add DNA polymerase, okay, we add DNTPs, we add buffering system, and we add maybe poly T primer. But to each tube, we add something different. Here, this something different is basically, to tube 3, we add primer that will detect, that will bind specifically to the cDNA synthesized from the blue primer. So we're going to call it primer B. To tube number 2, we add primer Y. And for tube number 1, we add primer P. So all 3 different tubes, they have the same tRNA. Let me add here number 6, total extracted our mRNA. So in each tube, we add the same 6 components, except in each tube, we add a different primer. So we have 2 types of primers in each tube. Poly T primer and a specific primer. So now we take these tubes, we put them inside the PCR machine. So we will have RT PCR. And now we have the 3 tubes. Okay. And then we analyze by gel electrophoresis. So after doing the RT-PCR, we will take, so here we have 3 wells that correspond to sample number 2, number 1. So we take, after RT-PCR, after we put the tubes inside the PCR machine or the thermocycler for a few hours, then we take the tubes out, and then we look for amplification of these genes. Okay. Let me add a tube here, a 4th tube. So tube 4 also will contain this mRNA, this mRNA, and this mRNA. But here I'm using a primer called primer double. Okay. And again, we have RT-PCR. We have 4 tubes. And on the gel, we have 4 different lanes to detect the presence of mRNA in the 4 tubes. So, primer P will allow the detection of the purple mRNA. If there is purple mRNA inside the cell, this means inside this tube there will be purple mRNA. This means that the primer will be able to detect the presence of the purple cDNA. Remember, here the purple mRNA, for example, will be converted into purple cDNA by the enzyme RTase and by the poly T primer. Okay. Well, let me use a different. Let me here use a different color. By the reverse transcriptase and by the poly T primer. But after that, using the PCR cycling and using the primer P, we will have multiple copies, billions of copies of the purple cDNA. Okay, so here we have billions of copies of the purple cDNA, and now these billions of copies could be, detected by gel electrophoresis. Same thing applies to mRNA Y. Same things applies to mRNA blue. Okay. So the idea here is that we're using the first stage of the RT-PCR to convert the mRNA into cDNA. This stage requires the RT-PCR, the poly T primer, and dNTPs, and a buffering system. The second stage will make millions of copies. Millions of copies. Yeah, Isra, this is the mechanism. This is the mechanism of converting the mRNA into cDNA. I just already explained. Okay. And then the PCR, primer P, dNTPs, the DNA polymerase will make millions or billions of copies of the cDNA. And these billions or millions of copies of the cDNA can be detected by gel electrophoresis. Okay, so if we found, for example, that we have a DNA band here, and maybe in 2 we have also a DNA band, in tube 3 we have also a DNA band, and in tube 4 nothing. Of course, the presence of a DNA band on the gel electrophoresis means that we were able to detect purple cDNA, which means this purple cDNA were made from purple mRNA, which means the purple mRNA was present inside the cell, which means the purple gene or gene P was expressed. So this one is expressed. Okay, let me here add gene W. Sorry. We found here in tube number 2, where we added primer Y. Primer Y will detect the presence of cDNA Y. We have a band, we have a positive result, which means the RT-PCR amplified the yellow cDNA. The yellow cDNA was made from the yellow mRNA. The yellow mRNA means it is present inside the cell, which means that yellow gene was expressed. We have a band here. Okay, of course these bands, they do not, on the gel, in reality they do not have different colors. It's just I use different colors to these bands just to indicate which gene we are looking at, or we are trying to decide whether it's expressed or not. And here, having a band, this means we have billions of copies of blue cDNA. Billions of copies of blue cDNA could be made from blue mRNA. The blue mRNA means which means that the blue mRNA was found inside the cytoplasm of the cell, which means that the blue gene or gene B was expressed. In the last lane, we do not see any band. This lane corresponds to tube number 4. In tube number 4, we added primer W. And primer W was not able to amplify any cDNA, which means there was no white mRNA inside this tube, which means there was no white mRNA inside the cell, which means that gene Y was not expressed. Okay. So now we decided that we have expression here, expression here, expression here, no expression for gene Y. If we look at the size of the band or the thickness of the band, this is the thickest. Thickest means أخمل, أضخم. This is the second thickest band, and this is the thinnest band. So a thin band here means low expression because we started with only one mRNA molecule. A very thick band here means high level of expression because we have more copies of mRNA, of the yellow mRNA, than the purple mRNA. And here we have thickness that is in between this one and this one, which means that the level of expression of the gene B, as you can see here, in this example, I added here 2 mRNAs. This means that the level of expression of gene B is in between Y and P. So if I want to order the level of expression, here we have the highest level of expression, followed by gene B, followed by gene P. So high level of expression is observed for gene Y, intermediate level of expression observed for gene B, and the lowest gene expression was observed for gene P. So not only we can decide which genes is expressed or not, yes or no question, but also we could use RT-PCR to have an idea about the level of expression. It's not an accurate estimation, but we could have an idea based on the thickness of the bands. Okay. So this is answering the question, which genes are expressed inside the cell. But also, we could use the same technique to answer a question about, let's say, when is the gene is expressed. So for example, if we have 6 developmental stages, okay, 1 to 6, and we extract mRNA from cells at each stage. Okay. So here we have, let's say, and we are interested in detecting the presence of the expression of gene B in each of these 6 stages. So here we're looking at the expression of a single gene, but at different developmental stages. So here we're asking when gene B is expressed. So notice here we use specific primers. This is the specific gene that we are looking for. Okay, this is gene B, let's say, and we use specific primers for gene B. So we extract mRNA from stages 1 to 6. Then we do RT-PCR. So we will have 6 samples, 6 tubes. Each tube will contain the mRNA from cells of that stage. And in each tube, okay, so we will have tubes, 6 tubes. And in each tube, we will add DNA polymerase, again, reverse transcriptase, DNTPs, primers, both specific primers and poly-T primers, and buffering system. But in each tube, we will have different mRNA sample. So here we have mRNA from stage 1, mRNA from stage 2, mRNA from stage 3, mRNA from cells of stage 4, mRNA from cells in stage 5, mRNA from cells of stage 6. And now we do RT-PCR for all these 6 tubes and we look at the result. As you can see here, we have a band from tube containing mRNA from stage number 6. We have a band from the tube containing mRNA from stage 5, 4, 3, 2, and there is no band where we added the mRNA from cells in stage number 1. So this means that gene B is expressed in stages 2, 3, 4, 5, and 6. Okay. But as you can see here, we have very narrow band, very thin band, which indicates here we have at stage 2 the lowest expression level. Okay. Here we have similar expression level. So we can conclude from this gel that gene B is expressed in embryonic stages 2, 3, 4, 5, 6. Its expression begins at stage number 2 and continues to be expressed at a high level in stages 3, 4, 5, and 6. Okay. We could also study the expression of thousands of genes at the same time. And this is by using what we call a microarray assay. So a DNA microarray assay compares the gene expression of thousands of cells or all cells inside, or the expression of all genes inside the cell in different types of cells or at different times or under different conditions. This is very important. So here we're trying to answer the question where, when, but for all genes, not only a single gene like here. Here we're looking at the expression of a single gene. Here we're talking about the expression of all genes, studying the expression of all genes at the same time and different conditions here also if. So the microarray, let me add a slide here. So what do we have in the micro? In the microarray, we have a glass slide, let's say. Okay. And on this slide, we have what we call, these are spots. Okay. So on each spot, we have DNA, single-stranded DNA molecules that are found in each spot. Well, maybe I should use different color. Let me use a different color. Come on. Let me use this color. These are single-stranded DNA molecules of the same sequence. So each DNA molecule you see in each spot corresponds to a specific gene. So each spot corresponds to a specific gene. Okay? This is for gene Y, Z, W, A, B, C, D, R, T, X, S, and R. So here we have genes called Y, Z, W. Each spot corresponds to each gene, and the DNA in each spot is a unique, consists of a unique sequence for each gene. So we have here, so the microarray consists of a tiny amounts of many single-stranded genes fixed to a glass slide. Okay, so the DNA that you see here corresponds to. So here we have single-stranded DNA, identical. Let's say identical. Oh, come on. Identical single-stranded DNA molecules corresponding to each gene. So here we have DNA molecules for gene T. Here we have hundreds of thousands of DNA molecules for gene R. Okay, we have double R. Let's call this Q. So for each gene, we have a specific spot, and we have single-stranded DNA molecules corresponding for each gene. And then the small DNA, single-stranded DNA molecules are fixed into a glass slide like this. Of course, here I'm showing you spots for 12 genes, but actually these glass slides might contain spots for all human genes. So some glass slides, they might contain, let's say, 21,000 spots corresponding to 21,000, let's say, human genes. Okay. For simplicity here, I'm showing you just only 12. So let's imagine that we want to compare the expression of genes, of these genes, inside, let's say, cell A. These are mRNAs. And we have here cell B. These are the mRNAs. Okay. We extract all the mRNAs from cell B. We extract all the mRNAs from cell. So these are mRNAs. These are mRNAs. So here we're trying to compare the gene expression between two different types of cells for these genes. And then we convert all these mRNAs into cDNAs. So here we have cDNAs let's say mRNAs from cell B, cDNAs from cell B. Same thing applies here, using RT-PCR. Using RT-PCR, we convert all the mRNAs into cDNAs. So here we have mRNAs A, here we have cDNAs from cell A. Then we add a fluorescent dye to cDNA A, let's say, so that now all cDNA from cell A will become blue, or let me use red. So here we have cDNA A, and then we add a fluorescent dye for cDNAs from cell B, so that all the cDNAs from cell B, they will become green. So we give the cDNAs from cell A a red color, the cDNAs from cell B a green color, and then we mix them together, mix the cDNAs and add to the slide. So what will happen is that we denature the cDNAs, so now we have single-stranded cDNAs. We mix them together and we add them to the slides. Some slides, now we will have hybridization. So for example, we have, use a different color. So we have here some cDNA from the red cDNA bound, hybridized to the DNA found in spot Q, here 2, here 2, maybe here. Because here we will have single-stranded cDNA, single-stranded DNA molecules. So complementary sequences will start hybridizing to each other like this. Also, we could have green, 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 and some green here. Only green. Okay. So now, as you can see, some of the green cDNA was able to hybridize to certain spots like S, D, R, B, Z, Y, and C. Some of the blue cDNA from cell A was also able to hybridize to the DNA in spot Y, C, T, Q. Notice that T and Q and Y, these spots contain DNA that is complementary to some of the cDNAs coming from cell A. Cell D and B, they have spots D and B. They have single-stranded DNA molecules that are complementary to cDNA coming from cell B. Spots R, C, and Y, they can bind both cDNA from cell A and cDNA from cell B. Okay? So now, if we look at these spots under the mic, from the top, what you will see something like this. We have 12 spots. Here, this spot will be red, red, red. Then we have this spot will become green, 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 green. Okay. And then we have spots that will have a color in between green and red, which is orange. So here we have orange. Let's say here we have orange. Here we have orange. Okay. So now, spots that are colored completely red means that these spots, they only hybridize cDNA from cell A, which means that the genes corresponding to these spots. So here we have spot Y, D, A. Sorry, this is X, Y, Z, B, R, S, U, T, C, and W. So gene the hybridization between a cDNA and the DNA on the spot means that gene is expressed. So red spots, if the color of the spot becomes red because of the binding of the red cDNA, this means that gene X, Q, and T are expressed in cell A only. Green spots means that genes C, B, Z, and D, they are only expressed in cell type B. Yellow spots or orange spots indicates that we have, like Y, we have both green and red cDNA binding here. So this means that gene Y is expressed in both cells. So here we have Y, R, C expressed in A and B. Black spots. The DNA in black spots were not able to hybridize to any cDNA, whether it's green or blue or red, which means here we have spot A and spot W not expressed in any of these, not expressed in A or B. Okay. So as you can see here, the color of the spot indicates whether a gene is expressed in cell A only or cell B only, or expressed in both cells, or not expressed in either cells. Okay, so we have 4 cases. XQT expressed only in cell A, SBZD expressed only in cell B, YRC expressed in both cells, A and W not expressed in A, neither B. Okay, so this is how to compare the differential gene expression between 2 different types of cells.